So our next speaker is Professor Manoj Gopalkrishnan, who is the founder and CEO of Algorithmic Biologics.
Hello everyone, good morning. It's a pleasure to be here at the symposium on big data algorithms for biology. And I'm very happy to follow after Kishore from Illumina. One of the reasons that this sort of ties into the previous talk in a very nice way is, so even if you look at the title of this conference, we are talking about big data and then algorithms. What Kishore was talking about in his talk was once the data is there in front of you, right? So once you've used Illumina's sequencing platform and you have all this data, what can you do with it? What algorithms can you apply to it to get biological insights? Yeah. My talk is going to be a bit more upstream of that problem. So I'm going to talk about how to get that big data out. Yeah. That's not a solved problem, even though companies exist, like Illumina and so on. There are various technologies out there from PCR to next generation sequencing. Getting molecular data out at scale, at an affordable cost, and accurately, that remains a very challenging problem today. In India, if you think about it, right, out of all the newborns who are born, there is a mandatory test called newborn screening, which detects for inborn errors of metabolism. 2% newborns are getting that test. If you think about people getting transplants, they are supposed to do something called an HLA test to know whether there will be rejection or not. There are a huge percentage don't get that test just because of cost. And so there are various such examples. Blood bank, right? Blood banks will go for the cheaper test like immunofluorescence instead of going for the molecular diagnostic test, because it's a more expensive test. Yeah? And this is not just in India, you can see this internationally. You can look at neonates who may have some infection, who may have sepsis, and within a 6-hour window they need a diagnosis of not only what is the infecting agent, but also what should be the path forward. Which antibiotic, should they be given an antibiotic? And there are still deaths in NICUs all over the world because of conditions like this, because we still don't, this is an unsolved problem, how to get the big data out. And I am here to tell you that algorithms have a big role to play in this challenge, not just once the data is out, but in the test tube and getting the data from the test tube to a scale where you can look at it. Yeah? I'll start with a brief introduction. I see many familiar faces here, but also many new faces. So my own journey has been, through academia. My PhD was in undergrad and PhD were in computer science. But for my PhD, I was in this field of research known as DNA computing, where you build computers out of DNA molecules that work inside a test tube. So I was working in a lab. So I was designing DNA nanostructures. I was building them. I was looking at them using atomic force microscopes, running gels, running PCR. So very much, in the lab. After my PhD, I have, been in academia for a few years and stepped out 4 years ago to start Algorithmic Biologics. The DNA of Algorithmic Biologics itself is on the intersection of molecular computing and statistics, or statistics, machine learning, AI, in my mind, equivalent words. Yeah, our team has grown over the past 4 years, and the core technology that we have been building is a compression technology that works inside test tubes. So before you pull information out, the information is inside the test tube in starting from some sample of biological origin. And what you want out of this testing is you want a result or a report, which will then feed the algorithms that many in this room are building, right? So this is the, the quality, the scale, the cost of this will decide what gets built, will decide what biological applications get solved, what diagnosis gets given to patients, what drugs get discovered. Yeah. So to get from here to here, we think of this process as some kind of a channel, and then we think of how to optimally use that channel, and the lessons that we have learned from information theory and coding theory come into play. And the key lesson here is that the best way for you to do this is to do something before you read the information out. You should do something inside the test tube and then do your readout using various kinds of technologies, and then you should do a decoding step or a decompression step, and we have software for that. So Algorithmic Biologics has built this stack. We have built a stack to do compression and a software stack which runs on the cloud to do the decompression. And I will give you a brief glimpse. It's a short talk. We have 20 minutes. So I'll give you a very brief glimpse of some of the use cases that we have covered. I am around today and tomorrow. Happy to chat with anyone who wants on sort of to dive deeper. Yeah. Okay. So so let me say one more thing before I go to this slide. So we do this compression in two ways for two kinds of situations. So one situation in which we do this compression is population level studies. Yeah. So suppose you want to diagnose people with rare disease in a population, right? So then you may have an ambition of testing an entire country for rare diseases, right? That's tens of thousands, hundreds of thousands, millions of people, right? So in those kind of situations, how do we do a compression? Yeah. And in those kind of situations, we are able to achieve compression simply by pooling samples together in various combinations. And I'll tell you about that. Yeah. The other kind of situation that we concentrate on is sort of closer to the clinic. There is a single patient who has come, and so single sample. If you have a single sample and you want to get information about that single sample, possibly a very large number of targets, so then we have an assay platform we have built based on molecular computing ideas that I will tell you about next. Yeah, and both of these platforms are starting to come together, and that is a story I won't have on this slide, but happy to chat with somebody who's interested. Yeah. Okay, so this is the first use case where we are looking at something like a population level screening, a large number of samples. So on the left, I am showing you a large number of samples. Here I have chosen for illustration a number 900. Yeah, but this could be smaller, this could be larger. There is quite some room to play here. These samples could be extracted DNA, extracted RNA, right? Various kinds of things. So these samples are now pooled, and these are my pools. Each pool, as you can see, contains a mix of various samples without any barcoding, without any other kind of processing. It is just a pool. Yeah, you know how to do these pools, right? I think it's just mixing things together. Yeah. Once we have these pools, the thing to note here is there is a pattern here. Every sample participates in multiple pools. It's not just that one pool contains multiple samples. And when one sample participates in multiple pools, the effect we achieve here is as if we have multiplied a matrix by a vector. So there is a vector of what we are trying to measure here that gets converted to another vector, a much shorter vector, which is now encoding the information that we are interested in. And this vector gets measured through whatever experiment is happening in the lab. And then there is a software here which is able in certain situations to recover the information here in a lossless manner. And what those situations are, when it works, I will get into some details as we go through examples. But the thing to remember here is this is a single round method. It can give you quantitative results per sample, and not just on a single target, but if you are doing PCR and some multiplexed PCR, you get a multiplexed result. If you are doing NGS, you can do whole exome, whole genome, you can get the entire sort of genome level results of variants, SNPs and indels and homozygotes and heterozygotes. You can get all that information out. And we have shown in studies that we have concordance with individual testing across various kinds of platforms. We have made the whole thing easy to do. So this is fast and easy to carry out. It's happening in partnership with commercial labs. It's happening in the real world. Yeah. And this has been proved for various platforms. So the same software is cross-platform and works across various kinds of technologies. Yeah, so so this is the sort of big picture idea. What a pool achieves is some kind of a summation effect. Yeah, so if you have a certain number of targets of interest here across the different samples, the number of targets of interest in the pool will be the sum of the number of targets here, or an average depending on how you take volumes and so on, with some noise term. Yeah? And so essentially every pool is acting like one row of a matrix multiplied by your target vector. Yeah? So with multiple pools you get the matrix vector product, and once you get the matrix vector product, the algorithm comes into play there. Okay, so we have done this for next generation sequencing. We, out of the top 5 genomics labs in India, we are working with 4 of them. We have done studies on whole exome and clinical exome level. and we are also internationally, we have just started in the US, we have just started with some early pilots. We are able to show with this method that while reducing library preparation by more than 60%, we are able to detect the low prevalence variants, so variants that are present in 5% of the population or fewer. So not all the variants. We are not promising we'll catch every common variant, but we are catching all the low prevalence variants. And we are able to do this with a very high PPV, and we are also able to call inserts, deletes, as well as homozygotes and heterozygotes. Yeah. Another surprise sort of when we were doing this is that we are able to do all this, saving not just library preparation, but also sequencing costs. So when we take our sequencing data for the pools, which was the same size as the sort of concordance, it was a concordance study, so we generated about 700 GB of data for samples. We generated the same size of data for pools. Yeah. So per pool, since there were smaller number of pools, per pool we were generating more data. Yeah. So we took that data and we started randomly throwing out reads. Yeah. We wanted to see how many reads we can throw away and still get a comparable sensitivity. So we were able to throw away 20-25% of the reads and still not lose any variance. Yeah. And even here, as you see, the numbers are going down on like the third decimal point. So we are literally losing like 1 or 2 variants even after throwing almost half the reads away. Yeah. So this has a capacity to reduce sequencing cost dramatically, and since then we have formalized the argument. We are together with one of the leading NGS labs in India, we are working on a manuscript that we hope to put out soon, which will explain the arguments and some of this data. Yeah. This is still an early preview that you are seeing here. Okay, so we have also done this for mass spectrometry, completely different technology. Again, the idea is the same. You have a large number of samples, you pool into small number of pools, you get data for each pool, and then there is an algorithm that comes in, is able to do a deconvolution step, sparse regression step really, to be able to identify, in this case, who are the heavy hitters, who are the individuals with a, with a much higher than usual amount of signal. Yeah. And this we did it for the use case of newborn screening for inborn errors of metabolism. And this is the first stage of this clinical study is done. The second stage we are currently in progress. We are doing this. Yeah, so we started off with qPCR during COVID times. We started off with campus screening. So during COVID times, there was a need to do asymptomatic screening, people who didn't have symptoms, and in those kind of populations we were expecting a very low prevalence, say about 5% of people would have COVID or fewer, and in those situations, in a small number of tests, we were able to give a large number of individual results while correctly identifying the individuals who had COVID. So this is 4 years ago, this is where the company started, but the same algorithm has since been extended to mass spectrometry and to newborn screening. Yeah. So this is one part of my talk where I spoke about situations where we are doing various kinds of population screening. The other part of my talk concerns an assay platform that we have built, and this assay platform, using this assay platform, we are able to achieve the same kind of matrix vector products that I showed you via pooling, but we are now able to achieve this for a single sample. And the vector now is the vector corresponding to the amounts of various targets that we are trying to measure, and the matrix vector product is achieved through this through this assay platform that we have built. Yeah. So the constituent of this assay platform is something we call sequence transduction. So A here can be DNA or RNA, sequence of interest, right? And so this could be SNP that you want to look for. This could be RNA from some gene expression level that you're interested in, or this could be some pathogen, right? So if you're testing for tuberculosis, you want to look for a certain pathogen or AMR sequence, right? As of today, if you want to detect a sequence, you throw in primers for it, you do PCR, right? Or you do sequencing and so on. What we are doing is we are able to rewrite the sequence using DNA computing, molecular computing ideas, to a completely different sequence X of our choosing. So this is essentially, you tell me what A you want here, what X you want here, I will build you an assay to convert A to X. Yeah? So that's what we are able to do, and the promise is, if X is detected, then A was present, if X is absent, then A was absent, and this is quantitative. So the amount of X tells me how much A was present. Yeah, and A can be DNA or RNA, so we can build in a single assay, we can build something to detect both DNA and RNA simultaneously without requiring a step to convert the RNA to cDNA. And this X is DNA, so we can do a downstream detection with any technology that detects DNA. So this could be PCR, this could be mass spectrometry, this could be next generation sequencing, melting curves, whatever you like. Yeah, the power of this this assay platform comes because it is highly multiplexable. I can send one target to multiple downstream proxies. So this A can go to X1, X2, X3, X10. Multiple targets in the same tube, in the same reaction, can be sent to multiple downstream proxies in parallel, or I can collapse multiple targets to be represented by the same single proxy. Yeah? And because I can do all these things, I can now do various interesting things. So I'll show you 3 examples that we have built in our lab, which will give you a sense of the power of this assay technology. Yeah? And please let me know on time. So one of the powers of doing something like this is that you can build a universal PCR. So you can build a PCR once and for all, and no matter what targets you want to detect, keep using the same PCR. Yeah? So imagine a diagnostics company whose inventory contains just one PCR, but they are able to build assays for whatever they want. Yeah? So here we built a PCR detecting sort of 4 synthetic targets. We made these up, and they all had the same end region, so that with a single primer pair, we could amplify all these 4. Then we took 4 E. coli genes, simply because we wanted something to test, and we transduced them to the corresponding PCR targets. Yeah? And then we did our reactions where we added some of these targets and not the others. And across all these reactions, whenever something is present, you see a CT value. Whenever something is absent, no detection. And across repeats, you see a very tight clustering of the quantitation, showing that it's a very clean PCR as well. Yeah? So this is one example where, here we used E. coli genes, but in principle it could be something else, right? Once we built this, we said, can we send 27 genes to the same target? Yeah? Can we do that collapse that we spoke about? And so we did that in the next experiment, where we took 27 different E. coli genes to that first target, used the same PCR, right, on the first channel. And not only did this work, when nothing was there, no CT, when something was there, we saw a CT, but it in fact worked like a summation. So the amount of output, this is all on a sort of logarithmic scale, right? CT values are on logarithmic scale, but the amount of summation as far as the qPCR quantitation accuracy goes, it is reasonable to think that this behaves like a summation. It's not a proof, but it is consistent with this behaving like a summation. And because we understand the underlying molecular mechanism, we have a strong reason to believe it behaves like that. We are actually working on a digital PCR platform now to verify that this behaves like a summation within much tighter quantitation bounds. Yeah? And once we did this, we said, can we do 27 more on the second PCR channel, 27 more on the third PCR channel, all in the same test tube. Yeah? And we did that. So we got a 3-plex, 81 targets in groups of 27 reported on 3 different PCR channels. Yeah? And once we did this, we said, now can we do our matrix vector product in some kind of a combinatorial way? And so we did that. So we took one sample, 243 different targets. These are again 243 different E. coli genes. And we did this kind of a matrix vector product. This is now a proxy. Yeah? And this proxy is the same as the first amplicon for the PCR, what I was calling EGFP1. Yeah? And this proxy is downstream of multiple targets. This proxy is downstream of a different set of multiple targets. Each target is going to multiple downstream proxies. Yeah? So this sets up again some kind of a code word, the presence of these proxies. Each proxy is informing me about multiple targets, and each target is encoded in multiple proxies. Yeah? So I get a matrix vector product here, and this is a 9-tube assay. Yeah? In tube number 1, I have EGFP1, EGFP2, EGFP3 as my 3 proxies, and the 4th one for an internal control. Tube 2, again the same 3, EGFP1, EGFP2, EGFP3 internal control. Tube 9, again the same things. The mapping is different in every tube. Yeah? In every tube, the same proxy represents completely different set of targets. So once this is done, I did 9 PCR reactions and got 27 numbers, 1 CT value for each proxy in each tube. by running exactly the same PCR reaction, literally, right, 9 times. The PCR was not different. I used only one primer pair across these 9 tubes, only 4 Tachman probes used across these 9 tubes, and I got 27 numbers which were fed into my software, and the software then did a sparse regression to be able to determine exactly which of these targets are present and how much. Yeah, provided no more than 5 targets are simultaneously present. That was the requirement for sparse regression to work robustly. Yeah. So in some sense we have been able to show a massive multiplexing in situations that resemble neonatal sepsis, when a small number of pathogens or small number of AMR markers might be expected to be present in a small number of reactions. Yeah. So we took this concept to various, so we see ourselves not as a diagnostics company, we see ourselves as a tools company. We build the technology, we don't have expertise in necessarily the regulatory pathway and going to market, which is quite challenging in diagnostics. So we took this to diagnostics companies, asking them, would you want to partner with us? Can we build something for you, take it to market? And the answer we got from them is, this is fascinating, but for us, the biggest problem that you would be solving with something like this is how fast you can build the assay. And so what they told us is, when we throw in hundreds of targets and try to build an assay for hundreds of targets, it takes us a long time to build that. It takes us literally 2 years simply to do the analytical validation. This is because when we throw in hundreds of primer pairs, we run into problems of cross reactivity, which leads to false positives. We run into problems of competitive amplification, which leads to false negatives, and we have to do multiple design, measure, learn loops to solve these problems. Yeah? And what we could do for them is essentially in 3 months, down from 24 months, we can finish the analytical validation, because the way we build assays, we have only one primer pair, so we don't have a problem with cross reactivity, and we are reusing the PCR, so we don't have a problem with this kind of competitive amplification, right? So we are able to reuse components, and we are able to solve cross reactivity. So the big picture story then was that there is something modular here. Just like you use an op-amp in electronics, you should be able to reuse a PCR when you are doing this kind of testing. And we have been running pilots for various diagnostics companies now internationally, and so we are in an early stage where we are building these assays from NIPT to minimal residual disease in cancer to infectious diseases like tuberculosis and so on. Yeah, so this is a picture to kind of take home, that if you have something that is good at detecting and giving results for universal proxies, you can make this a module in your assay design. And this is the hard part. So you build it once, you reuse it every time, and this part you kind of reuse every time, and this part is software, you reuse it as and when required. So that was my talk. Thank you. And if there are people in the audience who are looking for internships, please reach out. I'm here today and tomorrow. If anybody wants to come and chat, wants any more details, please reach out. Would love to talk and happy to take questions.
Thank you. Thank you, Professor Manoj. So,
very fascinating talk. I think you'll revolutionize diagnostics, in times to come. So the elephant in the room is contamination. So have you tested, how tolerant are these algorithms for contamination?
Yeah. So contamination is a multifaceted issue which has been plaguing the diagnostics industry. Yeah. And one of the ways in which contamination happens, in fact, we also have some IP here. Yeah, one of the ways in which contamination happens is that, say you are doing PCR, right, and you are testing, say, for COVID. Yeah, and what happens during PCR is you are amplifying something, and whatever you have amplified, it increases in number so much that if that is not disposed of properly, or even if sometimes if it is disposed of properly, it can get everywhere. It can get onto clothes, it can get onto bench, it can get onto door handles, and then the next time somebody is doing an experiment, those amplicons get into your reaction and you report a positive even when there is no positive. Yeah? So the advantage of doing things this way is that this step doesn't have an amplification, and this is swappable. So if there is contamination and you are seeing a negative control come out as positive, yeah, that is usually, we always run controls when we run diagnostics. So if a negative control comes out as positive, you don't need to stop the show and then do the whole decontamination and so on. You can swap out your universal proxies. You can directly go to a different set of universal proxies, immediately run flavor B of that reaction, and that will work. Yeah. So there are some unique ways, very different ways of thinking about contamination that emerge from this kind of point of view.
Lovely talk.
Thank you.
Lovely talk. I love the second half. In the first half, can you just go through the PKU example again? You mentioned you had 256 N, and then you just pooled them together, right? If you go one step before, for the, oh no, this was it?
Yeah.
So how did it happen? Like, you just pooled them, and then just please help me understand. This is the loveliest part.
Yeah. So we have done this in two different ways with two different labs. Yeah. So these are all dried blood spot samples, right? And so these dried blood spot samples, you can take a punch out of those samples, and you can pool at the punch level before you even do the extraction. That is one way that we did it, and this data that I am showing you was done that way. Yeah. Sorry. No. So how do we deconvolve? So the thing about mass spectrometry and inborn errors of metabolism is that first of all, it's a very low prevalence situation, right? So 1 in 1,000 is a high prevalence there compared to, most diseases are much more rarer than that. Yeah. And so in this low prevalence situation, the individuals who have a disorder show accumulation of a metabolite because the body is unable to get rid of it. And that accumulated metabolite is at a much higher level than the normal population, so that even in a pool, I get it as a heavy hitter. And then just because a pool is positive, I don't know who it is, right? So that would then waste all the effort because I would have to open out all my pools. Yeah. But because I have this kind of a combination, I'm able to triangulate and identify the sample. Yeah. Thank you.
What about some other metabolites or something which
So,
are slightly more, not so widely different?
Correct. So there will be limitations of this method. If a metabolite is very close to the normal range and we don't have that kind of resolution, we will lose it. Yeah. But so currently we are doing a phase 2 where we are doing some 43 different metabolites, and all of these, the normal range versus the threshold that is put, there seems to be a sufficient gap that we believe we should be able to call it, with a high.
Manoj here.
Yeah.
So I understood your first part of the talk, but I guess the second part was confusing to me because if you go to the signal transduction part, the reason is that for biologists it has a completely different meaning, so I got confused.
Yeah, so it's a sequence transduction,
I
not
understand.
signal.
So what I want to get a clarification, if suppose for example I have a mutation that I want to detect in 100 different samples. The sample is unique here, but mutation is not. The location is not. Now, how do you have like a design of a universal primer that I have to attach to my DNA before I go there, or that part I didn't understand.
So are you looking for a known mutation with a known
I'm
sequence?
looking for a known mutation, but 100 different samples. So here A is my sample, so I have A1 to A100.
Yeah.
Now when you change it to X1, does it mean that once I isolate my DNA, do I have to add a universal primer to all DNA?
So sorry, I think it's going to the other slide. Yeah, so A would be your mutant sequence.
Yeah.
Okay, so even though you have 100 samples, it doesn't matter. I'm only looking at one sample at a time. I have the same, there is no pooling here at all.
Yes, I have 100 samples.
Doesn't matter, I just will do it 100 times for each sample. So let's just focus on one sample.
Okay.
Yeah, so for that one sample, you have a sequence A. Yeah, instead of designing primers for that A, because see, you have A and you have A star,
Yeah.
right, which is the wild type, which is one base apart.
Yeah.
Yeah. If I convert A to X and A star doesn't convert to X, my job is then reduced to detecting whether X is there.
So
And
this
that I am able to
So
do.
this is the part which I got confused. So I'm still running 100 independent reactions to detect. So
Correct.
where am I?
Where are you
actually
gaining? Where are you
saving-
gaining? Yeah. So now if you tell me for that one sample that you
Yeah.
want to detect 100 mutations,
No.
I can do that.
No, I am detecting same mutation in 100 samples.
Yeah. So again, right, there are two different use cases here.
Yeah.
One is large number of samples.
Yeah,
One is one sample, large number of targets. Yeah?
correct.
Assay technology, as shown here, is one sample, large number of targets.
I see.
Pooling technology, as shown here, is many samples, one target, or possibly many targets. Yeah. So that is the pooling technology. So you may want, once you have an assay, any assay, your existing assay, you may then want to try the pooling method. With the pooling method, we will be able to tell you which samples have your mutant and which samples don't have your mutant in a smaller number of reactions.
And in this case, am I attaching a known like primer which is
You are not attaching, it's your assay. Only thing that is happening is pipetting,
Okay.
nothing else. After you are done pipetting, you run your assay here.
Right.
Your assay, as you run it, no changes.
Okay.
The data goes to our software. Our software then triangulates and says which of these samples
So
contain the
is there a lower limit in terms of the number of samples I must test to get this validated?
So depends on what you are trying to achieve. Yeah. So if you are trying to achieve compression, yeah, then if you have 900 samples, you get 90 pools. If you have 36 samples, you get 18 pools. If you have 9 samples, you get 9 pools. Yeah. So no compression at 9 samples. But even in 9 samples, 9 pools, or even in smaller than that, there are other advantages. So you get a triplicate testing of every sample. Yeah. So even if there could be an error in one of those tests, if it is done 3 times, you get that redundancy. So you are depending on what you want. So for example, in the NGS situation that I showed here, right? So in the NGS situation, even if I don't save on library prep, even if I run a very small number of samples, I will still get a savings at the sequencing level. I will be able to generate much less sequencing data and still get the same sensitivities.
Thank you.
So there is an accuracy boosting also happening here.
So, yeah, one last question.
This is just for practical application I'm asking. So you're saying that if we consider we have a panel of genes we want to test out for a particular disease, they can be done all at once using one sample, many targets.
Correct.
Yes.
Correct. So what I showed here, you know, in the POC, yeah, we were able to test one sample in the lab for 243 targets in 9 PCR reactions.
Okay. So the second part of question for this RNA, like RNA degrades faster. So in this, when we divide into different, say, like we don't call it aliquots, we call it in different parts. Does it affect, like this degradation affects this process?
So RNA degradation is a function of RNase presence, right? And so how well you are able to get rid of RNase, and so yeah, I mean, I have seen examples where RNA has been like heated at 95 degrees for like half an hour, 1 hour, and nothing has happened to it. Right? It's not as unstable as people fear, but RNase presence is the concern here. Yeah? So if a lab is already used to working, so my point would be, if you are trying to pool RNA, right? If a lab is already used to working with RNA, it should be fine. They should be.
And this is true for small RNA also?
It's true for small.
Short
it is
genes.
mostly small RNA. Yeah,
Okay.
it should be fine. Yeah.
Okay, thank you. So let's thank Manoj once again for a wonderful talk.
So sorry, question. I think you're talking about transcriptomics. We would not probably use pooling for that. We would. I'll talk with you. We have a different approach for that. Yeah. Yeah. Thank you.